My name is Kentaro Arikawa from SOKENDAI, Japan. First of all, I would like to thank the organizers for making this symposium actually happening in this difficult time. It is a great honor for me to be invited as a speaker today. Today, I will talk about the color vision of butterflies. The study of insect color vision is pioneered by Carl von Frisch more than 100 years ago. He studied honeybees and found that honeybees can see ultraviolet as a color, but not red. Indeed, honeybees do not visit red flowers very often. But insects are diverse. Many butterflies, like this papilio, they visit reddish flowers to take nectar quite frequently. They can probably see red as we do. So I thought that the visual system of the butterfly and the honeybee is somewhat different. This was the start of my study. This butterfly is the main figure of my talk today. I will divide the talk into 3 parts. First, I will explain how we study color vision in an animal like a butterfly. Second, I will introduce what sort of eyes do they have. They have compound eyes. And third, I will correlate the compound eye structure and color vision by referring to the dimension of color vision. Let me start from the first part. If you plan to study the mechanism of color vision in an animal, the very first thing you have to do is to check whether the animal really has color vision. The only way for this is to directly ask them through behavioral experiments. We first took newly emerged butterflies and forced them to take sucrose solution on a paper patch like this. In a couple of days, they become able to visit the disc and take sucrose solution when hungry. It is now drinking sucrose solution on the disc. Then we presented 4 discs of different color on the floor and tested the trained butterflies. This time we did not put any reward on any discs. This particular butterfly, trained to the red disc for 3 days, correctly visited the red disc, even when we changed the position. This butterfly was trained to visit the blue disc, as you see. The same thing happens when we train them to green or yellow discs. Okay, it seems that the butterflies can discriminate 4 colors, 4 colored discs. However, this is actually not good enough to demonstrate their color vision. Suppose that they look these discs in black and white. The discs can still be different in the brightness as you see in this picture. The butterflies could have used the brightness difference when making a decision. To exclude this possibility, we did another set of experiments where we placed 1 colored disc with 7 discs of different shades of grays. If the butterfly sees the discs in black and white, it would confuse, for example, these 2 discs that look similar in brightness. However, such a confusion never happened in any butterflies. All the trained butterflies visited the disc of the trained color. We can thus conclude that the papilio butterfly has color vision. Our color vision system is made robust by some additional functions, such as color constancy. What is color constancy? Here's a red apple. The apple appears red under the sunshine or under the light from a bulb. The reflection spectra from the apple must be quite different in these two lighting situations because the radiance spectra of these light sources are different. But still, we say that this apple appears red. This is due to our color constancy. Color constancy is actually crucial as well for animals that discriminate objects by colors. We tested color constancy in Papilio. This yellow-trained butterfly visited the same patch on the color Mondrian under the reddish illumination. This pair of pictures already indicates that they have color constancy to some extent. We also demonstrated some other functions, such as simultaneous color contrast, the lightness contrast, or polarization vision in Papilio. I will not go into all the details about these things today, but this must be an interesting direction of color vision studies in animals. Okay, let me explain a little bit of textbook information here. What is light? Light is a small fraction of the electromagnetic wave. The electromagnetic wave, whose wavelength is between 400 and 700 nanometer, is visible for us, the human visible light. The light shorter than 400 nanometer is called ultraviolet light, which is invisible to us, and the light longer than 700 nanometer is called infrared, which is also invisible. We see lights of different wavelengths as different colors. This is due to our color vision function. As you saw in the butterfly experiments in previous slides, color vision can be defined as the ability to discriminate visual stimuli based on the chromatic contents independent of the intensity. So, why can we see the lights in different colors? The fundamental mechanism exists in our eyes. In the eye, we have the retina. In the retina, we have many light sensors, which are called photoreceptor cells. Each photoreceptor has the spectral sensitivity. This graph shows the photoreceptor spectral sensitivity, plotted against the wavelength. There you see three different curves. This one has the peak in the blue wavelength region and therefore called a blue cell. And this one had a peak in green, and this one had a peak in the red wavelength region, therefore they are called green cell and red cell respectively. If the light stimulates only the blue cell, we sense blue. But the light stimulates the green cell and the red cell at the same time, we feel yellow. This is the physiological basis of our color vision. By the way, why the spectral sensitivities are different among these photoreceptive cells? Here I have to refer to a molecule known as the visual pigment, or rhodopsin. A visual pigment molecule consists of a protein called opsin, and a vitamin A aldehyde 11-cis-retinal attached to it. Light is absorbed by this 11-cis-retinal, and light converts the 11-cis-retinal into all-trans-retinal, which leads to the photoreceptor to be activated. The amino acid sequence of the protein determines the color of light absorbed by the 11-cis-retinal, and we humans have 3 different kinds of opsins in our eyes, which absorb blue or green or red, respectively. These different opsins are expressed in one of these types of photoreceptor cells, making these specific spectral sensitivities of these cells. Now, how about in butterflies? How many color sensors do they have, and how are these sensors organized in the eye? Here, let me move to the second part of the talk, the compound eyes. As in most insects, the eyes of Papilio are compound eyes. This is a picture of a Papilio compound eye, which consists of about 12,000 small units called Omatidia. A single Omatidium is a slender, very long structure with a lens and a bundle of photoreceptors inside. If you cut the compound eye through this plane, you will see something like this. This is a cross-section of an Omatidium. These flower petal-like things are the cell bodies of photoreceptor cells. These photoreceptors together make a photoreceptor site in the center, which is called the Rhap Dome. The Rhap Dome consists of the visual pigment containing cell membrane. Cell membranes are rich in phospholipid, a type of oil in the body. From the optical point of view, oil has higher refractive index than water, and therefore the Rhap Dome acts as a light guide or an optical fiber. At any rate, a single Omatidium has a single photoreceptive site, the Rhap Dome, which means that an Omatidium corresponds to a single pixel of the image formed by the compound eye. Next, let's think about what the insects might be seeing. Some of you might know this toy called the compound eye lens. People say that you can experience the insect's visual world by looking the scene through this compartmentalized lens. This is how you see a single flower through the lens. Each flower image corresponds to a single compartment of the lens. Is this really true? Is that what the compound eye sees? Let's have this small portion enlarged. In this enlarged picture, you see 9 by 9 pixels, 81 pixels. We see this complex image or complex shape of the flower because of the numerous pixels contributing to it. However, as I showed in the previous slide, a single Omatidium corresponds to a single pixel, and a single pixel cannot form any images. So, playing with this lens is fun, but this image with multiple flowers is clearly wrong. Then how do they see the world? Here I compare the retinas of a butterfly on the left and humans on the right at the same magnification. This is 20 microns and this is 20 microns. In the human retina, each dot corresponds to the photoreceptor, and the colors indicate blue, green, and red cells. The white circle in the center corresponds to the image of 1 yen coin put about 1 meter away from you. The image covers the area of about 90 photoreceptors in humans, but in Papilio it covers only 1 Omatidium. In other words, the pixel density is much higher in humans. What is determined by the pixel density is the visual acuity. We can roughly predict the visual acuity of other animals by using this principle. Here I show you some examples. The scene we look like this should be something like this in cat, whose visual acuity is 0.17, and for Papilio like this, and the fruit fly, a tiny little fly you often find in your kitchen, is like this. The visual acuity can be estimated as 0.003. The compound eyes look pretty hopeless. However, I will explain that the compound eyes are in fact not hopeless at all. They are super. Again, I show you this picture. An Omatidium contains several photoreceptors, making the Rhap Dome in the center. Here, this section was stained with a blue dye called Azure 2. Therefore, this section looks blue. If you bring the section under the microscope before staining with Azure 2, you will see something like this. Here is an Omatidium, and here is the Rhap Dome in the center. You see, the Rhap Dome is surrounded by 4 reddish dots, and the color of the dots is red in here, but yellow in here. And this has red pigment, and this has yellow, red, yellow. So it seems that there are 2 different types of Omatidia according to the color of the pigment surrounding the Rhap Dome. I have to make a long story a bit short here, and we found that these pigments act as color filters for the Omatidium. If you eliminate the compound eye from the inside, you will see the Omatidia look in bright red or yellow. Of course, the red Omatidia have red pigment inside, and the yellow Omatidia contain yellow pigments inside. And here are some pink Omatidia, and they have another characteristics which can be seen in this slide. These 2 pictures were taken from the same sample. The left one was illuminated from inside, as in the previous slide, and the right one was taken under the UV illumination from above. You see some Omatidia emit strong fluorescence, bluish fluorescence under UV illumination. If you superimpose these 2 pictures, you will see the fluorescing ones correspond to these pinkish ones. These simple experiments have revealed that the compound eye of Papilio contains 3 types of Omatidia. The first type has red pigmentation, and the second type has fluorescence pigment and the red pigment, and the third type has yellow pigment. These 3 types of Omatidia are distributed rather randomly over the eye, in the eye. Okay, under the next, let's see the structure of the individual Omatidium. An Omatidium has the cornea and the crystalline cone lens at the top and the photoreceptor layer here. The photoreceptor layer contains 9 photoreceptor cells numbered 1 to 9. These photoreceptor cells together construct the Rhap Dome in the center. The arrow indicates somewhere wrong. It should be in the center here. And the distal of the Rhap Dome is actually two-tiered. The distal 2/3 of the Rhap Dome is made up with the photoreceptor membrane of the photoreceptor number 1 to 4. And the proximal 1/3 is made up with the photoreceptor number 5, 6, 7, and 8. And photoreceptor number 9 is a very small one, adding a little bit of photoreceptive microvilli at the very bottom of the Omatidium. And the next question is the sensitivity, spectral sensitivity of these photoreceptors. To measure the spectral sensitivity of individual photoreceptors, we employ single-cell electrophysiology, where we prepared a very fine glass micropipette filled with some dye inside. Then we penetrated the electrode into a cell, stimulate the cell with a series of monochromatic lights, and recorded the response. And after recording these responses, we injected the dye into the cell to mark it. As a result, we can get a set of results, something like this. This is a green sensitive cell peaking at 520 nanometer, and here is the dye injected cell, which corresponds to R4 corresponds to this cell in the diagram. And I will show you a couple more examples. Like this red receptor peaking at 600 nanometer was the R8 photoreceptor in a red pigmented Omatidium. This is a violet receptor peaking at 400 nanometer, and which was found in this particular Omatidium, which emitted fluorescence under the UV illumination. Okay, we repeated this sort of experiments many times, and finally we could identify several different photoreceptors. And in addition to the blue, green, red cells like in our own eyes, we found a UV receptor, a violet receptor, and a broadband receptor, which covers very wide range of the wavelength. We of course identified the locations of these 6 spectral receptors. And this slide summarizes the results, the location of these cells. For example, type 1 red Omatidium contains UV, blue, and red receptors, and yellow pigmented Omatidia contains blue and green, and so on. This table shows a bit more detail. For example, type 2 Omatidium occupies 25% of the Omatidia, has red pigmentation and a fluorescence under UV. And it contains violet receptors and green receptors and broadband receptors. These letters in red show the visual pigment molecules they express, but I will not go into the details about these molecular biology today. Okay. Rather, I would like to introduce you the situation in some other butterflies. Papilio was the very first butterfly species whose eyes were studied in this detail. Then a question arises, of course. Do other butterflies have the same eyes? The answer is, of course, no. This is a small white butterfly, Pieris rapi, Monshirocho in Japanese, which is very common worldwide. And this is the compound eye. This is the compound eye of Pieris with about 6,000 Omatidia. And the eye is also a collection, appears a collection of a few different types of Omatidia, as shown in this picture. We carefully analyzed the anatomy and physiology of the photoreceptors and identified 8 classes of receptors, which are very different from those of Papilio. I was very much impressed by the discovery of 2 types of red receptors, the red and dark, which are very close to each other. Another surprising point in the small white is the sensitivities are sexually dimorphic. The violet receptor is found only in females, and the males have double peak blue receptors instead. The males and females see the world, probably world, very differently. This is the result of the common blue bottle Graphium salpidon, Aosuji ageha in Japanese, and I'm pretty sure that you cannot even count the number of graphs in here. There are actually 15 spectral sensitivities in here. 15 is the maximum number known in one insect eye. In this species, however, we did not find any sexual dimorphism. A prominent sexual dimorphism was found in the eastern clouded yellow, Collia serrate, Monkicho in Japanese. Males have 6 classes of receptors, while females have 8 classes of receptors. Females are clearly rich in the variety of red receptors, which may be beneficial for finding good quality leaves to lay eggs on. Okay, and here is another example, the Asian wood white Leptidia amirensis, Himeshirocho in Japanese. And this butterfly is close relative to the small white butterfly. And we studied the eye of this species as well, and the eyes are not spectrally complex. It's very simple with only the UV, blue, and green sensitive cells, like in honeybees. And the eyes are spectrally simple, but we noticed another complexity in this species. And this is the compound eye of the Leptidia amirensis. And you see that the ventral part, the lower part of the eye, looks dull. When I first saw this, I thought that this region had been damaged somehow, but actually it was not the case. And under the scanning electron microscope, we found the eyes are actually rough, appears rough like this. And this is the high magnification picture, which indicates that there are small Omatidia and large Omatidia sitting side by side. Even more interestingly, the roughness of the eye is found only in males. This is an eye of the female Leptidia amirensis, which is quite smooth. And we measured the areas of the corneal facet lenses in both sexes. What we found was that males have two populations of Omatidia, while females have only one population. Again, the strange sexual dimorphism indicates that the visual world can be sexually dimorphic. When we were thinking about the biological function of the eye roughness, a colleague of mine told me about this unique digital camera, which is no longer on the market. It has a particularly expanded dynamic range with a specific type of CCD. The CCD of the camera has small and large pixels like this. The small ones has lower sensitivity shown in here, which starts responding to the light of fairly high intensity and saturates at higher intensity. And the larger pixels have higher sensitivity, start responding at very dark light and saturates rather quickly. And the overall sensitivity of the whole CCD should be the summation of these two sensitivity curves, which is the black line here. And this black curve has the expanded dynamic range from each of these single pixel sensitivities. We thought the similar thing might be happening in the Leptidia eyes. Then we measured the overall eye sensitivity by placing a special ring electrode over the eye and recorded the electrical responses to the light stimuli of various intensity and measured the overall sensitivity of the eye. This graph shows the intensity dependency of the response in females and males. And the width of the dynamic ranges was compared between the 10% response and 90% response. As you see, males, which have the rough eye or two different omatidial sizes, have expanded dynamic range compared to the female. The dynamic range is about 0.5 log unit wider. The 0.5 log unit difference is actually huge. In the real environment, the 0.5 log unit difference roughly corresponds to the difference under the direct sunshine and the dark shade here. Males of Leptidia amirensis are known to fly actively in and out of the shade, but females tend to stay rather close to the food plant, which are often found in the shaded places. This has to be directly studied, directly checked in the field, but I have a feeling that this is a promising hypothesis. Okay, we have learned that not all butterfly eyes are created equally. The structure and function of compound eyes must be closely related to their lifestyle. Here, let me come back to Papilio, and the question now is, how do Papilio butterflies use their complex compound eyes to see colors? I would like to address this question from the viewpoint of the color vision dimension of color vision. And as I explained, Papilio eyes contain at least 6 classes of spectral receptors. An obvious question arising here is whether or not the butterflies use all of these receptors to see colors. In other words, is Papilio color vision hexachromatic? There could be various ways to answer to this question. And the way we decided to take was to measure the wavelength discrimination function, or the delta lambda function. This graph shows the delta lambda function of humans. This curve shows the discrimination threshold, or the minimum discriminable wavelength difference, delta lambda. So delta lambda 1 means that we can discriminate only 1 nanometer difference in the wavelength range around here. So this means that if we see 600 nanometer and 599 nanometer light side by side, we can tell the difference. So the curve has 2 lower places, which are high sensitive regions. So these existence of these 2 troughs is because our color vision system is trichromatic. At around 500 nanometer, the spectral sensitivities of blue cell and the green cell overlap, and at around 600 nanometer, the sensitivity curves of green cell and red cells are largely overlapping. So the number of troughs indicate the dimension of color vision. If we can measure the wavelength discrimination in Papilio, we can therefore estimate the dimension of their color vision. This is the setup we made to measure the wavelength discrimination of Papilio. The light coming from the xenon arc, the xenon lamp, is reflected by this mirror into the monochromata, and the resulting monochromatic light was passing through the ND filters and reflected up again by this mirror, shining the window on the benchtop experimental bench from below. So the butterfly can see the light on the top of the bench through the window. And we have the same set of optics on the right-hand side to make the presentation of 2 independent lights possible. We first trained a butterfly to take nectar at the light of certain wavelength. In this case, the wavelength is 480 nanometer. The trained butterflies extend the proboscis here, the proboscis towards the light. You see? Again, I will show it again. It extends the proboscis toward the light to take nectar. This training is actually not very easy. Only very good butterflies can perform well. Using the successfully trained butterflies, we tested the wavelength discrimination by presenting two lights. One is always the light of training wavelength, and the other window was illuminated by some other light of the wavelengths close to it. If the butterfly can discriminate these two lights, it extends the proboscis towards the training wavelength. This shows a typical set of results obtained from the individuals trained to 480 nanometers. When the test wavelength, the horizontal axis shows the wavelength of the test light. When the test light wavelength is 480 nanometer, the butterfly is seeing two identical lights, 480 nanometer. So the choice frequency of the training wavelength is around 50%, like that. If the wavelength of the test light is becoming shorter or longer, the choice frequency of 480 nanometer increases, making this curve typically in V shape. If we take the 60% at the threshold in this case, we can measure the wavelength differences they could discriminate. In this case, 1 nanometer or 2 nanometer in this region. And we did this sort of training at 16 different wavelengths and obtained this wavelength discrimination function. As this slide shows, this graph shows, we could find 3 clear troughs at 430, 480, and 560 nanometer, suggesting that the color vision system of Papilio is tetrachromatic. The performance of Papilio is actually quite good. This is the curve of humans, and the minimum discrimination threshold is around 1 nanometer, which is equivalent to that of Papilio, but the Papilio has wider coverage, expanding into the UV wavelength region. This is the graph result measured in honeybee back in 1972, and it has 2 troughs, because the color vision of honeybee is trichromatic based on the UV, blue, and green receptors. And I'm quite glad to see that the color discrimination is the best in Papilio in these 3 species. We still don't know which 4 out of 6 contribute to the wavelength discrimination. To get some insights into this point, we created a mathematical model, the noise-limited color opponency model, based on the intrinsic noise of photoreceptors and the number of each photoreceptor types. I will not go into all the details of the equations here, but the basic idea is that the more photoreceptors exist, the weaker signals can be reliably detected. and we calculated the wavelength discrimination function using all the 6 receptor types in here. The thick curve showed the result, and this is not bad in the longer wavelength region, but here is a huge discrepancy in the UV region. And we ran the model calculations and finally found that the model gave the reasonable fit when we excluded the violet receptor and the broadband receptors from the calculations. So this suggests that the papilio color vision system is based on the UV, blue, green, and red receptors. Okay. So they have UV, B, G, R tetrachromacy. This is again the Omatidium array of papilio, and we found that the violet and broadband receptors are not necessary for color vision, maybe not necessary for color vision. And both are in the second type, type 2 Omatidia, which are probably used for some other visual functions, for which we still don't know, like shape vision or brightness vision, whatever. Let me conclude the talk by showing this take-home message slide. I talked about the papilio eye, and it has 6 spectral sensors in the eye and has tetrachromatic vision. And although I didn't explain these details, we showed color constancy or color contrast or polarization vision, or the ability to see colors at the spatial resolution limit. And we also learned the species and sex, sexual dimorphism, sexual specificity in the visual world. And we are now actively working about the brain mechanism of color vision, and also the evolution of color vision system among insects. And I hope that you enjoyed the world of butterfly vision through my talk today. And I would like to thank these my lab members by introducing them to you, and also to you attending the symposium. Thank you very much.